Genetics is a relatively young science that has great potential to dramatically change the lives of all people on the planet. Genes play a crucial role in the life of every living thing. Recent scientific discoveries allow us to modify genes the way we want them to be. In this video, I will tell you about the history of genetics, the most important discoveries in this field, and what the future might hold. The discovery of genes and the role they play in heredity can be traced back to the work of Austrian botanist Gregor Johann Mendel in 1864. Mendel was a monk, but was very interested in the world around him and studied it with curiosity. He was conducting an experiment in breeding pea plants when he noticed that specific traits were passed down from parent plants to their offspring unchanged. For instance, when a tall plant was crossed with a dwarf plant, only tall offspring were produced. Through his experiments, Mendel discovered that hereditary information was passed down from generation to generation in indivisible units, which he called genes. This discovery unveiled the smallest building block of heredity and laid the foundation for our modern understanding of genetics. Years later, Dutch botanist Hugo de Vries expanded upon Mendel's work by merging his ideas on genetics with Charles Darwin's theory of evolution. De Vries discovered that genetic differences occur randomly in nature, known as mutants, which helped to explain why variations in traits exist in the first place. This work complemented Darwin's ideas about species evolution, showing how physical traits are transferred from parents to offspring through genes. Together, the work of Mendel, de Vries, and Darwin provided a complete picture of species evolution. Random variations in traits occur in nature, are passed down to offspring, and are naturally selected over time as some offspring survive while others die. The discovery of genes has revolutionized the field of genetics, paving the way for modern genetic research and the development of new technologies such as gene editing and personalized medicine. However, the implications of this knowledge also come with significant ethical considerations, and society must grapple with the consequences of genetic modification and manipulation. With each new discovery in genetics, it is essential to carefully consider the implications for human life and the ethical implications of our actions. The discovery of the gene marked a significant breakthrough in understanding heredity, but it also led to new questions about the nature of genes and how they work on a biological level. In the 1940s, biochemists began examining the workings of cells, and in the nucleus of the cell, they discovered DNA, deoxyribonucleic acid, and its close relative, RNA, ribonucleic acid. These molecules, grouped as nucleic acids, were found to consist of 4 components, known as bases. DNA's bases include adenine, A, cytosine, C, guanine, G, and thymine, T, while RNA's bases are identical, except for uracil, U, in place of thymine. The discovery of DNA finally provided scientists with the building blocks of genes. The traits observed by Mendel were found to live on an organism's DNA strands, and multiple genes work together to express observable traits like height. However, the relationship between gene expression and visible traits isn't always straightforward, as external factors can influence these traits as well. For example, a boxer's nose is shaped not just by genetics, but also by repeated encounters with hard-punching opponents. The role of the environment in shaping visible traits is an important factor to consider as we continue to unravel the complexities of genetics. DNA tells each cell in a body what it will become and exactly when to do so. Each cell in our body has a specific function, such as skin cells, muscle cells, or liver cells. But how do these cells know what function to fulfill? The answer lies in DNA, the genetic code that each organism begins life with. When the first cell begins to divide, DNA tells each new cell which role it should play in building the new organism. This process continues as cells divide and take on new functions. This leads to the creation of an embryo in 3 stages, starting with the definition of the main axis, followed by the activation of specific genes to form body parts, and finally the turning on or off of genes to form unique organs or elements of a particular species. However, DNA's role in cell function goes beyond telling cells what to become in a developing organism. DNA also controls the timing of when cells assume their roles. In a groundbreaking experiment in the 1970s, biologists at Cambridge University mapped every cell in a male earthworm, predicting which role each cell would play and exactly when it would do so. They discovered that cells could even disappear at certain times in the worm's development because of the instructions encoded in its DNA. These findings illustrate the remarkable complexity of the genetic code and how it governs the development of an organism from a single cell to a fully formed individual. But history knows of cases where the science of genes has had very bad consequences because it has been used for evil purposes. The field of eugenics emerged in the late 19th century as a way to improve the human gene pool. Supporters of eugenics believed that by applying scientific methods to human reproduction, desirable traits could be increased in society while undesirable traits could be eliminated. One of the early proponents of eugenics was Francis Galton, the cousin of Charles Darwin, who believed that traits like intelligence, strength, and beauty could be selectively bred for in society. This idea of using genetics to purify humanity eventually led to the first court-ordered sterilization in the state of Virginia in 1927. Carrie Buck, who was deemed feeble-minded by the court, was sterilized based on the logic that it would prevent her from producing feeble-minded children and protect the population from undesirable genetic traits. However, it was Nazi Germany that took the concept of eugenics to its terrifying extreme. Adolf Hitler and his administration used the ideas behind eugenics to justify the elimination of groups deemed undesirable, including Jews, Gypsies, and disabled people. The Nazis distorted genetics to enact a policy of systematic sterilization, arguing that Jewishness and Gypsiness were the result of hereditary traits. By 1934, nearly 5,000 adults were being sterilized monthly. The Nazis then went on to commit wholesale exterminations of these groups, with a final death toll of around 11 million people. This regime demonstrated to the world how genetics could be twisted for nefarious purposes, leading to eugenics becoming a taboo field that remained so for generations. In the 1970s, geneticists made significant progress in the fields of recombinant DNA and gene sequencing. They sought ways to manipulate DNA more quickly than the natural slow process of recombination, which requires multiple generations. Researchers wondered if they could speed up the evolutionary process by creating new genetic combinations in a lab. Biochemists Paul Berg and David Jackson from Stanford University showed that it was possible to create new organisms by inserting the entire genome of a virus called SV40, together with 3 genes from the bacterium E. coli, into a lambda bacteriophage. They called their new creation recombinant DNA, a process that became known as gene cloning. The question then was whether it was possible to read DNA instructions. The precise order in which bases appear on a DNA strand determines genetic information, not the bases themselves. Gene sequencing seeks to read genes by knowing their sequence or the precise order of the bases. In 1977, biochemist Frederick Sanger became the first person to successfully sequence a genome or complete set of genes. He mapped all 5,386 base pairs of virus Phi-X174. This discovery led to the continued development of gene sequencing and illuminated new aspects of the language of DNA. Researchers found that in animal DNA, a string of base pairs is often separated by long stretches of stuffer bases, which don't seem to code for anything. Instead, they act as a space or pause between useful base sentences. Gene sequencing technology has advanced tremendously, and it can now sequence a complete human genome in a matter of days. This has led to various applications, including identifying mutations that cause diseases and providing personalized medicine. The information that can be gleaned from reading DNA is vast, especially when it comes to the diagnosis of genetic diseases. By analyzing a person's genetic information, scientists can pinpoint the location of improperly functioning genes, which can serve as markers for potential disease. For instance, Down syndrome can be diagnosed during gestation, before a child is born, since individuals with this genetic syndrome are born with an extra copy of chromosome 21. In contrast, certain genetic diseases are much harder to identify since the cause of the disease is not a single gene or extra chromosome. One of the diseases that is difficult to pinpoint through genetic analysis is cancer, which results from the cumulative malfunction of dozens of genes in a cell. Since cancer is also genetically diverse, even two cases of the same type of cancer could be caused by entirely different gene mutations. For this reason, a genetic diagnosis of cancer requires a review of the patient's complete genome. This need for a comprehensive understanding of the genome was the impetus behind the Human Genome Project, which launched in 1990. The Human Genome Project aimed to sequence the entire human genome, which consists of more than 20,000 genes. At the project's outset, the idea of a complete human genome sequence was unimaginable to early geneticists, but advances in gene sequencing technology made it possible by the 1990s. In 2000, the project published the first draft of a complete human genome, and by 2003, the map of every gene in the human genome was made available online. This milestone marked a major achievement in the field of genetics, and provided a powerful tool for the diagnosis and treatment of genetic diseases. Now that we have access to a complete map of the human genome, what can we learn from it? The Human Genome Project was an international effort to map the complete DNA sequence of human beings. With this breakthrough, geneticists could now research every single human gene and compare the genomes of people from around the world to trace the origins of the species. One major discovery is that all humans share a common ancestry, with the oldest human populations found in the San tribes of South Africa, Namibia, and Botswana, and the Mbuti pygmies of the Congolese Ituri forest. Genetic analysis also shows that racial categories are a human invention and that they don't reflect any inherent biological differences. Research has revealed that less than 100,000 years ago, all humans lived on the African continent, and certain groups migrated to other parts of the world. For instance, white Europeans are the descendants of a group that migrated out of Africa around 50,000 years ago. Such genetic diversity can't explain why one race is superior or inferior to another, and scientific claims of inherent racial differences have no basis. Genetic variation takes millions of years to occur, and more importantly, some 85 to 95% of genetic diversity in the human species is contained within racial groups. Thus, two individuals from different parts of Africa are actually more genetically different from each other than they are from people of other races. Genetic analysis has shattered many myths about race and human diversity. Instead, it has helped us trace the origins of our species and gain a deeper understanding of human biology. As individuals, we often use various identifiers to define ourselves, such as our nationality, religion, or social class. However, when it comes to gender, the majority of people still identify as either male or female. This distinction is determined by genetics, specifically one particular gene. Your sex is determined by your 23rd pair of chromosomes, regardless of whether you're born anatomically male or female. For females, the two chromosomes are identical and are called XX. For males, one of the chromosomes is shorter, resulting in a pair called XY. Research has pinpointed the SRY gene as the key marker in sex determination. Discovered by Peter Goodfellow in 1989, an active SRY gene increases the likelihood of being born anatomically male. Gender identity, on the other hand, is a much more complex issue. Many individuals identify as something other than the traditional binary genders of male or female. Some identify as neither, while others identify as both. In reality, gender identity is non-binary, and it makes genetic sense. While an active SRY gene may determine anatomical sex, it doesn't directly affect gender identity. Instead, the SRY gene influences dozens of secondary genes that react to environmental factors to determine an individual's gender identity. As a result, genetic factors vary widely, leading to a diverse range of gender identities. The nature versus nurture debate is a bit more nuanced than proponents of either extreme will admit. The bottom line is that people are born with tendencies and not traits, and it's only when these tendencies interact with the environment that they become visible traits. In a 1979 study, behavioral psychologist Thomas Bouchard followed identical twins who were separated at birth and raised in entirely different environments. While the siblings had an identical nature, they had nowhere close to the same nurture. The experiment found that twins raised in different environments exhibit the same behavioral tendencies, although their exact behavior is ultimately different. In a particular case, one male twin was raised as a Nazi youth while his brother spent summers on a kibbutz. Both twins defended their beliefs rigidly and with passion, despite the belief systems being opposed. Similarly, since tendencies need to interact with the environment to become observable, a person with a predisposition toward violence will only act violently when exposed to violence, for example in the home. But the environment does more than just solidify tendencies. It physically etches itself into our genome, which is the subject of a field called epigenetics. When a gene is activated or deactivated by an environmental cue, like a traumatic experience or captivating smell, small molecules called methyl tags attach themselves to a gene. These tags act like annotations to a cell's DNA, somewhat like comments in the margin of a text. As tags accumulate over time, they begin to influence the functions of the cell. The result is that environmental experiences can affect how our genes are expressed, and that in turn can affect our behavior, cognition, and the potential applications of advanced genetics to improve human life are vast and promising. One of the most exciting areas of research is gene therapy, which aims to cure diseases by inserting genes into a sick patient to alleviate symptoms. Although the concept of gene therapy dates back to the 1980s, its development has been hindered by the danger of inserting viruses into humans. Scientists have since made significant strides in developing safer and more effective methods for gene therapy. Another area of genetic research with enormous potential is stem cells. These versatile cells can regenerate or transform into any other type of cell in the body, allowing scientists to manipulate genes and create genetically modified organisms from scratch. For example, researchers have successfully created a mouse that glows under blue light by inserting a jellyfish gene into stem cells extracted from the rodent, which were then mixed with embryonic cells and inserted into the womb of a female mouse. This promising research could lead to new treatments for a wide range of diseases and conditions. As our understanding of genetics advances, so too does our ability to harness its power to improve human life. While there are still many challenges to be overcome, the potential benefits of gene therapy and stem cell research are too great to ignore. With continued investment and development, these exciting fields could revolutionize the way we treat disease and improve health outcomes for people around the world. The understanding of genetics has the power to transform humanity in many ways, but with great power comes great responsibility. One area where genetics could have a huge impact is in disease diagnosis. Schizophrenia, a mental illness that is difficult to diagnose, is caused by a series of genes scattered throughout the genome. However, as sequencing techniques improve, doctors may eventually be able to diagnose schizophrenia in utero. But with advances in diagnosis come ethical concerns. For example, mental illness and creativity are often closely related, and many people with mental illnesses have gone on to lead inspiring, creative lives. This raises the question of whether it is ethical to abort a fetus that is likely to develop a mental illness. Is it acceptable to judge the value of a potential life based on genetic fitness, as the eugenicists of Nazi Germany did? Another area of concern is genetic modification. Scientists are already on the way to producing the first modified human. The process involves genetically modifying human stem cells before converting them into sperm and egg cells, which are then used to produce a human embryo through in vitro fertilization. The result could be a human resistant to all known blood diseases, or something even more remarkable. As we gain more knowledge and power in the field of genetics, we must grapple with these ethical questions and ensure that our use of this knowledge is for the betterment of humanity. What do you think about interfering with the human genetic code? Write your answers in the comments below. If you liked the video, please subscribe to our channel. It really helps us grow. See you soon.